Hi class, Dr. Jim here. In this lecture, we're going to be looking at all the eukaryotic cells. So last time we looked at the prokaryotes. They are very simple cells, not much to them on the inside. A lot of the fun stuff is on the outside. In this lecture, we're going to look at the eukaryotic cells. Now, hopefully you've had a class before that talked about the difference between prokaryotes and eukaryotes, like a biology class, or maybe even discussed it a little bit in your A&P. What we're going to look at today are the eukaryotic cells, and these are the cells like you and I are, or cells that have a nucleus. And so we're going to talk about those. And yes, there are cells that are like us that do infect us when we talk about things like protozoa, or we talk about fungi or molds that cause us to get sick, or maybe even something very large like a worm that can kind of get us sick in that stuff. And so we're going to talk about these different things today. So really the first part of this lecture is going to be looking at what a eukaryotic cell is. And then the second part, which I don't really care so much if you take a lot of notes on, we're just going to briefly go through and kind of look at a survey of these different things. We're going to be looking at some of the eukaryotic cells that actually can get us sick. And so we'll talk a little bit about that. And when we get to that point, I'll tell you, hey, you can put your pencil down, just kind of watch or listen and that stuff and kind of get an idea where we're going to go later on in this semester. So we'll talk a little bit more about that later on. Okay, so what are we talking about today? So the first thing we're going to look at is what are the eukaryotes and how do they come about? And so we're going to talk a little bit about the eukaryotic cell and then also talk about the thing called the endosymbiont theory, which is where the prokaryotic cells came together and formed a eukaryotic cell. And so we'll discuss a little bit about that. The second thing we're going to focus in on is what are the major structures of eukaryotic cells. So this will be hopefully a brief little review for you guys going through the major parts of the eukaryotic cell. So we'll talk about things like the Golgi, the rough and smooth ER. We'll talk a little bit about lysosomes and some of these things that you probably have heard before. If you haven't had biology, maybe an A&P or some other class that you may have taken somewhere in the previous. If not, hopefully this will be something that it won't be too difficult to grasp. And then finally, like I said, the second part of the lecture, which we'll quickly go through, is going to be the survey of some of these organisms that cause us to get sick. So again, I'll tell you to drop your pencils or do whatever. You don't really have to take notes on it. It's more of just kind of going through and looking at what are those things out there that can get us sick. So let's take a look at these different things and kind of get an idea of what we're talking about with eukaryotic cells. All right. So the first thing we have to think about is where do the eukaryotic cells come from? So if we think about back to chapter 1, I had that little diagram about the timeline of the Earth. And so it roughly states the Earth was formed about 15 billion years ago, and that the prokaryotes came about 4.5 to 5 billion years ago. The eukaryotes didn't come about until about 2 billion years. And so there was a long time, for about 2.5 billion years, that there were only prokaryotic cells and no eukaryotic cells around. Well, a lot of this things that were happening were due to what the prokaryotes were doing to the environment. And again, when you have life, life causes things to change. And so we hear a lot about now global warming and what humans are doing to the environment and everything else. Well, the prokaryotes did a lot to the early environment that made a lot of changes to cause the eukaryotic cells to actually come about. And so one of the things that happened was that you had these photosynthetic bacteria that were able to use sunlight, and one of their byproducts was producing oxygen. And so when you had more oxygen in the atmosphere, that atmosphere changed and it caused the organisms to change with that. And so you could kind of say that this was a global change in the environment, and that we're looking at the production of oxygen. And so one of that brought about changes in how these bacteria behave and how they work together. So a couple of things that happened, and I'm going to zoom in on this diagram here and kind of talk a little bit about the history of the eukaryotes. So the first thing that happened was that the DNA that was kind of floating around inside the prokaryotic cell became encapsulated in a membrane. Now, we see evidence of this in other cells, where in some of these prokaryotic cells, they actually do have some internal membranes, but they're not fully surrounded. What we're saying is over a long period of time that the eukaryotic cell developed this membrane, this bilipid membrane that formed around the DNA and formed the early nucleus. The second thing that happened was that these large prokaryotic cells, now that had their DNA incorporated by a nucleus, started swallowing up these smaller little bacteria that were able to use the oxygen as an energy source. These big guys were using anaerobic, and they knew that the environment was changing, And so they had to find a way to utilize the oxygen that was in there. Instead of eating these guys up and spitting them out again, what they did was they said, hey, come live inside of me. I'll take care of you if you are able to use the oxygen. And basically, that's what happened in this first set. And so we see the development of what we think is the early mitochondria. These were bacteria cells that were able to use the oxygen. The oxygen was then made to be able to make ATP. These cells would survive, and so you had this kind of symbiosis going on. These big, large cells protected these tiny little cells from being eaten by other organisms, protected them on the inside, while these little small organisms could utilize the oxygen that was going on in the atmosphere in making ATP and energy for the cell. Another thing that happened was that these cells then also ate up some of these little photosynthetic bacteria that were changing the environment. So some of these cells decided, no, we don't need them, and they became the animal cells, and so cells that are a lot like us. Other cells said, you know what, I'm really lazy. I don't even want to even eat. I'll just get my own food because I can do photosynthesis. I can make my own food. I have other bacteria that can make ATP, and voila, we can be really in charge and going in style. And that's what happened with the plant cells. So what happened is these large cells then that had the mitochondria then started to eat these photosynthetic bacteria. Now they could make their own food. They could make their glucose source, which then could do ATP, or basically go to cell respiration. Cell respiration could be done in the mitochondria, and voila, now you have a plant cell. So the animal cells, they have to still eat. They have to get their glucose from the outside. Whereas plant cells, they can produce it all by just sitting in the sun and absorbing the sunlight. And so that's how we think it originally happened. Now, what gives us any evidence of this? Well, if you look at the mitochondria and chloroplast, they both have DNA inside of them, which is kind of interesting. Why would an organelle need DNA? The second thing is they have 70S ribosomes, which you find in prokaryotes, not in eukaryotes. So that's another interesting thing. Also, if you look at the size of the mitochondria versus the size of the chloroplast, those are very similar to the size of the photosynthetic and the aerobic bacteria that are around there. And so there's a lot of evidence that really suggests that this theory actually makes a lot of sense. And so it's still being debated. It's not completely 100% supported, but there's a lot of evidence suggesting that this is how the eukaryotes came about 2 billion years ago. Okay, so now let's take a look at what's inside these eukaryotic cells. And so this video will kind of go through what I just talked about how these different things happened, how the environment changed and that stuff. Highly recommend. Again, it will be a link on the website, so you can just click on this after watching the video and you can go through, and it kind of goes through and explain it. Okay, so now on to the eukaryotic cells. So let's go on to the eukaryotic cells. So here are some of the ancient eukaryotic cells, shows you some of the differences in the chloroplast. So you can see the cells that had the chloroplast, and then also some of the cell walls. Okay, so let's look at these eukaryotic cells now. So these are the eukaryotic microbes. This includes protozoa, which are always single cellular. We'll also talk about the fungi and algae, which are multicellular or could be single cellular depending on how they are. And then we'll talk about the very developed worms. And then so you hear about the worms crawling in people and that stuff. And we'll look at all these things later on, either today or in the rest of the semester. Okay, so here's the eukaryotic cell in its glory. So again, most of the stuff now is on the inside. So when we talked about the prokaryotic cells, all the fun stuff was on the outside. In the eukaryotic cells, all the fun stuff is on the inside. So we're going to talk a little bit about what a mitochondria is, what a Golgi apparatus is, what a chloroplast is, what is the smooth ER, what is the rough ER, what are these centrioles and things like this, and talk a little bit about those different things. And again, this is hopefully review for you guys, so I won't spend a ton of time going over these things, but it is important to know the difference between a prokaryote and eukaryote, especially since we're going to spend most of our time on prokaryotes this semester. All right, so if we look at the organization of the eukaryotic cell, you're going to see that we'll first talk about the external structures, and this includes the appendages, and that's flagella and cilia. This is a little bit different than what we talked about before. Cilia are like fine hairs, and to give you an idea what cilia are, if you think about your respiratory tract, the trachea has cilia that move things around. That's what we're going to be talking about. We'll talk a little bit about the glycocalyx and eukaryotic cells. Again, it's not as important as the prokaryotes, but again, it does serve a purpose of, again, sticking to things, allowing you to stick to things. Then we'll talk briefly about the cell walls and cytoplasmic membranes. Not all eukaryotic cells have a cell wall. So we'll look at that as well. So like fungal cells and algae do, whereas the protozoa and animals and the worms do not. And then we'll finally get into the internal stuff. And again, we'll look at all these things very briefly and go through. Hopefully, like I said, this is a review, so we won't spend too much time on this. Okay, so the first thing we're going to look at is the flagella. And again, they talk about the structure. The structure is much larger than the prokaryotic flagella, about 10 times thicker and much bigger. The other thing is how it moves. So if you remember back to my lecture, I talked about the prokaryotic flagella spinning either clockwise or counterclockwise. Okay, and so depending on the way that it spins allows the bacteria to either move or move away from something. Well, in eukaryotic cells, this becomes more of a whip-like motion. So you think of the sperm cell making it to the egg, or maybe a tadpole swimming to the food or something like that. And so it kind of gives more of the snake-like or S-like pattern, and again, kind of lashes around like a whip. And so that's kind of how the flagella move. And so it's a big difference in the movement of how these flagella, not only are they thicker, but they move differently than the prokaryotic cells. Okay. Second thing that eukaryotic cells have are cilia, and cilia again are more of these long like hairs, and they look like little hairs all over the cell. And again, they kind of whip back and forth, and again, they can do a lot of different things. They can function in motility. You're talking about a paramecium moving through the water. They can function as a way to feed, so they move their food towards their mouth or their oral groove that they might have. And then finally, they might use it to filter. And so we use our cilia to filter things out. And so that's our filtering system. All the junk that we breathe in every day gets filtered through. When we sleep, we bring that stuff up to our esophagus, we swallow it, and then we wake up in the morning with kind of a frog in our throat. And the reason for that is all the junk that we were breathing all day that day before was kind of swept up by our cilia, and then we swallow it in the middle of the night. And so that's what you're kind of feeling if you got a lot of phlegm and congestion in the morning. That's all the gunk that you swallowed up. And it's a great way to clean yourself out. It just doesn't make you wake up very well and that stuff. You might have that really deep, sexy voice during the morning or whatever type of thing, or the really raspy voice. And so that's due to the cleaning or the filtering of the cilia. Okay. Here I have a nice little video. Again, I have it a link on the Blackboard page. So if you just click on it, you can see the difference between the flagella and cilia. And again, you can see the movement difference between the bacteria and the flagella. And what I'll do is you can look at both of them side by side so you can actually see the difference in how those guys move. So it just gives you an idea of how these things actually move in the eukaryotes. Okay, so another thing that the eukaryotic cells do have is a glycocalyx, and we've talked about this before. The glycocalyx is a sticky outside layer that allows them to stick to things, and so they can either have a slimy layer or more of a capsule-like layer, more complex, but typically, again, it's allowing for attachment to things. And so a lot of times you have these things to stick to different parts of the environment. If you're in the water and you want to stick to a substrate or something like this, if you're a parasite or something, so you can stick to things. And so again, you can talk about these things. And again, it's mostly for adherence, but it can be used for protection, sometimes for signal reception if you're signaling the outside world with multiple cells and that stuff. But typically what we see is underneath that glycocalyx, when we get under that, we see that the algae have a thick, rigid cell wall. And then the protozoa and animals lack that cell wall. So again, we don't see that they, have a cell wall in this case. So the glycocalyx is kind of on the outside, along with the cilia and flagella, and then now we're moving in. So before we spent a lot of time on the protozoa, or not a protozoa, but on the bacteria on the outside, talking about all these outside structures and everything. We're going to quickly move to the inside in the eukaryotic cells. All right, so the cell wall, again, only found in fungi and algae. The rest of the cells we're going to be talking about, the animal cells and the protozoa, do not have these things. But again, it acts as a layer of structure and support and protection for these cells. So again, it provides structural support and shape. The fungi have a polysaccharide fibers composed of chitin. And if you think of chitin is a polysaccharide that you have in the exoskeleton of insects. So when you hear a bug go crunch, that's the chitin busting up when you're squashing that bug. The algae have different things. A lot of times it's cellulose, very similar to what a plant cell has. And algae is very similar. And so what you'd be interested to hear about, any plants that are typically in the water, that is considered algae. They're not really considered plants. They're very similar in structure and size and everything else, but once they're completely in the water like that, they're considered algae. And so, again, we're not going to get into that much about algae and that stuff. That's more for biology. But for these things, again, you can see lots of different things that make up the cell wall, but these guys have it. So think of when they have cell wall, the fungi and the algae. Everything else in the eukaryotic world does not have a cell wall. All right, so inside that cell wall, we have the cytoplasmic membrane. We're going to talk more about this in chapter 7 when we talk about transport. So don't worry too much about this right now in this unit or this section. But again, it serves for transport, keeping things in or taking things out or moving things across. Again, you have a mosaic of a lot of different things. It's a phospholipid bilayer. We're talking about phospholipids in the next unit when we get into chemistry. So we'll talk more about there. We also have proteins that give some structure. The one thing I want you to think about is these things are fluid. They can move through the membrane all around the cell, and so nothing is structured or staying in one spot. Everything is very fluid, so it kind of moves along like a river would. And so think about it like that. Again, and then that contains all the cytoplasm on the inside. So let's move to the inside of the cell now. All right, so the first thing, obviously, that's different in eukaryotic cells than prokaryotic cells is the nucleus. Nucleus is basically where the genome is kept, and so the genome is quite different in eukaryotic cells. Typically, we talk about linear chromosomes as opposed to one bacterial chromosome that's round or circular. In eukaryotes, typically the DNA is linear, and you have multiple chromosomes, more than one, okay? Inside the nucleus, you typically have the bilayer. You have the phospholipid bilayer surrounding the DNA, protecting the DNA with lots of these little holes. These nuclear pores allow for the RNA, the mRNA to leave. And again, we're going to talk about that in chapter 9 when we talk about transcription, translation, and that stuff. But for now, just think of the DNA as in basically a big ball, or think of chromosomes on the inside. DNA transcription takes place inside the nucleus. Translation takes place outside the nucleus. You can also talk about the nucleolus, which is a dark spot in the nucleus. Sometimes you can see those when you stain eukaryotic cells, and that encodes for the rRNA. And we'll talk more about this again when we get into chapter 9. But for now, we'll keep it simple and just say nucleus to another membrane surrounding the DNA, protecting the DNA. You have the nuclear pores that allow for the mRNA to leave the cell. All right, and so again, we'll talk about mitosis, and again, going through the process of the mitosis itself. You have the 4 phases: prophase, metaphase, anaphase, telophase, and again, that's the dividing of the DNA. We're not going to spend any time looking at that. I'm not going to ask any questions about that. You probably had enough of this either in biology or A&P, but for the time being, you know that instead of binary fission, the eukaryotic cells divide by mitosis and going through that process. And we'll look at this again when we get into the farther chapters like chapter 22, 23, 24, and 25, when we look at all these more complex structures. All right, so other internal structures, we have the ER, and the ER stands for endoplasmic reticulum, and essentially they are production sites, and what they do is they produce things for the cell. We either have the rough ER, which produces the protein, so I always like to associate rough with ribosomes. Rough and ribosomes make proteins, and so the rough ER makes the proteins, okay? And so essentially it's these membranes that come off the nucleus, that where the mRNA comes out of the nucleus and then meets the ribosomes, you make the proteins there, and that's where the proteins are made. Okay, further on down from the rough, where you don't see any more ribosomes, you now have the smooth. And I think of slippery or oils, that's the lipids. And so the lipids are made in the smooth endoplasmic reticulum. And so again, the smooth ER is where the lipids or fats are made, in that case, storage and manufacturing of the lipids. Okay, and again, you can see the different structures. Here's the rough endoplasmic reticulum. It's rough because it has the little ribosome dots all over it. So here are the polyribosomes, and again, they're used to make the proteins. And you can see here, goes on in the ribosomes, the mRNA comes in, the mRNA now makes the proteins, and here comes the proteins off, and then they get let go into these membranes, which then gets circulated to the Golgi if it needs to. Okay. Here's the Golgi. The Golgi I always refer to as like the FedEx or the post office. And so if you need to get a package out of the cell, you're going to send it to the post office or FedEx so they can send it to wherever you need it to go. And so that's what the Golgi does. Basically, what it does is it takes these little vesicles that come off the endoplasmic reticulum and then converts those and changes it, does whatever it needs to do in order for the protein or the lipid to be sent out of the cell. And so that's what it does. It basically prepares, it manufactures, it gets ready to send out and transport whatever material you need to leave the cell. Okay, so it modifies, stores, and packages the proteins. And so, again, think of the post office or FedEx when you're talking about the Golgi. All right. And this is basically how this structure moves. And so if you think about it, the DNA is the source or the blueprint of the proteins. That DNA then gets transcripted into mRNA. That mRNA comes out to the ribosome where it gets manufactured. That's the translation part. The protein gets made. The protein is manufactured and then sent to the Golgi to be processed and packaged. Once it's processed and packaged, these vesicles then can be pushed out to the cell, and then finally you have secretion by exocytosis. So you can kind of think about it. You go from the nucleus to the rough ER to the Golgi to the vesicles, and then finally secreted out of the cell. And that's typically what happens when you're talking about these very large cells. These cells will do lots of different things. They work together and they secrete tons of different proteins and other things that other cells might need if you're talking about a multicellular organism. And so this happens all the time. It happens in you all the time, where basically you're making proteins for other parts of your body. Those things get made in one cell, they get secreted out, and then they go to another cell where then they react to whatever they need to do there. And so that's typically how these things happen. All right. So some other internal structures that are kind of membrane associated are lysosomes. Lysosomes are basically where things get eaten. They contain digestive enzymes that attach to the phagosome, which is what gets eaten inside the cell, and then digest those materials. And so we're going to talk more about lysosomes and phagosomes and all these different things when especially we look at the role of white blood cells. White blood cells do a very good job of eating things up that are not supposed to be there. And so we'll look really indiscreetly about the lysosomes and the phagosomes at that time. Vacuoles are basically membrane-bound sacs to store food or liquid inside the cell. So you talk about vacuoles all the time, storing things. And then here's the phagosome down here. And again, that's the vacuole, which basically the food is brought into the cell, or the pathogen or some of these other things that are brought into the cell. And then the lysosome and the phagosome combine, and then that's where the digestion or eating takes place. And so again, we'll look at these structures again when we talk about some of the things that go on in the immune system. But for now, just kind of remember these are some of the structures that are membrane bound that are also found inside the cells. They're not really organelles, but they play a role inside the eukaryotic cell. All right, so a couple of the other kind of organelles we're going to be talking about are energy generating. So you really can divide these organelles into two different groups. You can talk about the ER, the Golgi. These are just membrane bound organelles. Whereas we talk about organelles that actually produce energy. First is the mitochondria. This is the powerhouse. All cells have these things. And again, these guys are where cell respiration takes place. And we'll talk more about cell respiration in chapter 7 and 8 and looking at those things, how the whole process of glucose getting converted into ATP takes place. But essentially in the eukaryotic cell, this is where this process takes place. Inside the mitochondria, you have those membrane-bound proteins that do electron transport and generate a boatload of ATP for the cell. And so very important, we call these the powerhouse. And again, you can look at some of the different structures. On the inside of the mitochondria, you have the internal folds, which we call the cristae. This increases the surface area, more efficient, more ATP can be made. And again, we talked a little bit about some of these things. One of the interesting things is mitochondria can divide separately or independently of the cell. So these tend to divide during the S phase of mitosis, and so, or interphase type of thing. When you're synthesizing a new strand of DNA, these guys are independently replicating so that they can be divided up between the cell when the cell divides. It also, like I said before, when we talked about some evidence of these things, contains DNA and also prokaryotic ribosomes. And so that's important to notice as well. In eukaryotic cells have 80S ribosomes, the bigger ribosome. In the mitochondria, they have 70S. And this can be an important fact when we talk about antibiotics and that stuff. So kind of keep that in the back of your mind for a couple weeks when we get into the antibiotics, because I'll bring that up again and why sometimes when we talk about protein synthesis, it can be detrimental to our cells, even though we don't have the 80S ribosome or the 70S ribosome. So keep that in mind. All right, the other structure, obviously, that we don't have, but other eukaryotic cells do have, are chloroplasts. And chloroplasts are, again, used for photosynthesis. Photosynthesis is the process of absorbing sunlight, converting that sunlight from solar energy into chemical energy in the form of glucose. This is how plant cells or algae cells actually eat. And so this is an important process. If you've had biology, you've discussed photosynthesis. I know we've talked about this before. But if you haven't had a lot of photosynthesis, we're going to get a little bit into it in chapter 8. But for now, just know that this is where sunlight gets absorbed by the cells. The sunlight then gets converted through a process through light and dark reactions and basically gets converted into glucose. And we'll talk more about this in chapter 8. But again, this is where we have this process going on during or in the cells themselves, chloroplasts. Makes us different than plants. Plant cells have them. We don't. And so that's why plants are green. Green plants, they have chloroplasts. We don't have chloroplasts, so we're not green. All right. And then the ribosomes themselves, we talked a little bit about the ribosomes. I mentioned already in eukaryotic cells, these are bigger. Again, they have the two subunits, but in this case, we have the 40S and the 60S. In prokaryotes, they had the 30 and the 50. And again, the 30 and 50 make the 70S. Don't use math to add them up because, again, it's about how they spin. The 40 and the 60 add up to the 80S. And so they're different in eukaryotes. They're much larger in eukaryotes, a much larger size. And so, again, we see them either with the rough ER or they can be also free-floating in the cytoplasm as well. But a lot of times we see them with the rough ER, and we've talked about this already. mRNA comes in, you generate the polypeptide becomes a protein, and voila, we go on to that. And again, we'll discuss this more in both chapter 2 when we discuss proteins, and then again in chapter 9 when we talk about transcription translation, and we'll look at this again. But just remember, eukaryotes, larger ribosomes. Okay, then again, there's the cytoskeleton inside the cells of eukaryotic cells. We have a cytoskeleton, the actin and myosin. So you probably, if you've had AMP, you've talked about actin and myosin and how they can contract and expand cells and do these different things. And again, it's used for cell movement and a lot of different other types of things, transport, structural support. I don't talk a lot about it because it's not really the focus of this class. But again, you may have discussed it quite a bit more in AMP and looked at those things. So just to kind of give you an idea, that is for some of those structures. So it's the actin, myosin, the microtubules, and filaments that kind of give the support and structure to these cells. All right, so here you go. Here's the big list comparing the prokaryotes and eukaryotes. And I'm not going to go through all these things. I'm not going to bore you to death going through these different things. But again, some of the big things that are big differences that you see. Genetics, typically prokaryotes, again, they both have nucleic acids, but you have the nucleus in the eukaryotes, the nuclear envelope. You reproduce by mitosis in eukaryotes, where you do binary fission in prokaryotes. Again, the mitochondria is important for respiration, whereas in prokaryote cells, they don't have the mitochondria, so they do something different. Photosynthesis the same way. Again, shape and protection, the size. Size is really a big thing. Bacteria cells are really, really tiny. So when we get into lab in a couple weeks, when we do the gram stain, I'm going to have you guys do the gram stain of your gum line. You're going to see eukaryotic cells are huge. They're going to look like big fried eggs, and the bacteria cells are going to be these little tiny cells that either sit on them or are next to them and around them. And so you'll see the difference in the size. So eukaryotic cells much, much larger than the prokaryotes, about 100 to almost 1,000 times larger. So again, we'll talk about this. And we'll discuss viruses all in the next chapter. So hold on to that. We'll discuss those later on in the next chapter. All right, so let's do a concept check here. So let's see if you guys got anything from what I was just mentioning. So this question, it says, the eukaryotic organelle that is responsible for transporting vesicles inside the cells are what? The Golgi, the smooth endoplasmic reticulum, the rough endoplasmic reticulum, or the nucleus? I'll give you a minute to kind of think about this question. Okay, it is the Golgi. The Golgi is responsible for transporting the vesicles, and so that's what you think about in this case. The Golgi bodies or the Golgi apparatus is responsible not only inside the cell, but also outside, packaging and shipping things out of the cell. So there you go. Okay. All right. So Now you can put your pencils down. You can just listen to me talk for a few minutes. We're going to do a really quick survey at some of the different things that are out there that can get you sick. And so we're just going to take a quick survey talking a little bit about the fungi. We'll look at the algae, which are actually plants, or plant-like, I should say, the protozoa, which are single-cellular animals, and then finally finish up with the worms. And I'll talk a little bit about some diseases. Again, don't worry about it. I'm not going to throw this on the test. It's just going to be some general information and that stuff and just talking about it. So you kind of can sit back and kind of listen and just kind of pay attention to what's going on here with these guys. All right, so let's start with the fungi. So the fungi, there's over 100,000 species that are divided into two groups. And really, we consider the macroscopic. These are your mushrooms and puffballs and things like this, things you can see. And then you have the microscopic, the ones that get people sick, the yeast, the mold, sometimes the mildew, things like this. Those are the microscopic, the ones you have to use the microscope for, obviously. Again, most are unicellular or colonial, and few have some cellular specialization. Again, this is a typical agar plate that if you left out and let it to grow, you'd have fungal spores landing on it and growing lots of different types of fungi on it. So with the microscopic fungi, there are the two types. We have the two morphologies. You have the yeast, which you guys know about the yeast. Yeast grow and make bread, make beer, make wine, all these different things. And then you have the hyphae. The hyphae are the long fibers. And again, when you look at it from your point of view with your eyes, you see it's kind of furry. And the furriness is basically the hyphae. These are the hairs that you see. If you look at it in a microscope, you're going to see these long strands of fiber that are kind of growing throughout. And that is the hyphae, or basically the strands of fungi that are growing either on top of the media or below the media. And again, they can sometimes do both, but typically they're either one or the other. And again, a lot of times we see this with some pathogenic molds, and we'll talk a little bit about that as well. All right, so again, what is the nutrition? All have to eat. Now, instead of like animals that actually bring things into the cells, what fungi do is they excrete their digestive enzymes outside the cell, and then they kind of digest it outside the cell, and then they kind of slurp it back up again. And so it's kind of like if you had to throw up every time, throw up all your digestive enzymes, digest it out on your plate, and then take that plate and go and dump it in. Very nice picture, I know, explaining that. But that's basically what fungi do, okay? Most of them are harmless. Most of the time you see them outside. They're the mushrooms and that's some. Sometimes we even eat them and that stuff, and they're not, and they're actually pretty tasty, okay? But there are some that are parasitic in that they can live off of us, and so they cause these different things. And when they do cause disease, we call these things mycoses, okay? Mycosis, or mycoses, which are basically fungal infections. And you probably have had some type of fungal infection in your life. Maybe not, maybe you've been lucky. But if you've had something like this, where you've had dermatitis, which is basically a fungal infection of the skin. Sometimes you get it of the nails and in the toenails and that stuff, causing the yellow toenails and things like this. Other times you get the burning and peeling of athlete's foot. That can be another thing. Sometimes people can get it growing in the back of their throat, which is called thrush. And other times you can get it in places that you really don't want it, and especially in the reproductive organs and sometimes in the vagina, where you get the yeast infections and things like this. And again, fungi like to grow in warm, dark places. So wherever warm, dark places are on your body, you can get fungus to grow there. And so again, how fungus pass are through spores that are in the air. They land on material, and then they sprout and they grow. So if you keep yourself clean and and good hygiene, typically you don't have problems with fungal infections. But again, sometimes different things can change it. We'll talk more about this in chapter 22. But that's typically how people get it. A lot of times through skin-to-skin contact. You get sweat and other things. You don't change your clothes very often or things like this. You get the buildup of fungi. And we'll talk more about this as well, again, in chapter 22. Found in lots of different habitats as well. All right, here's the diversity again. Here are the microscopic fungi. You can see lots of different microscopic. Here are the mushrooms. And again, lots of different types of fungi that are out there. And then again, the roles of fungi. So again, obviously the adverse impact are the diseases, the mycoses. You have allergies. Sometimes those spores can cause and have you get itchy eyes or runny nose or things like this. So some of us suffer from seasonal allergies, and mold spores can be one of those things. Sometimes they cause toxins in production that get into food and make you sick. And so if you've ever heard the story about the witches of Salem, people thought they were witches because they were kind of crazy. But what happened was is mold got into the rye, and so the rye was used to make bread and other materials that they ate. This mold caused a toxin, and that caused them to hallucinate. And so it made them look like that these women were witches. But in fact, it was actually a fungal infection or a fungal intoxication from eating this fungal infested rye. And so it was actually not them being witches or possessed by demons, but it was actually due to this toxin eating this contaminated rye. Obviously, destruction of crops and food storages. You know, if you've ever had moldy stuff and you had to throw it out, you get mad because you spent money and your hard-earned money on buying these things in your refrigerator, and then it comes out moldy and that stuff, and now you got to throw it away. So that can be a bummer as well. Beneficial impact, obviously, they decompose the things that die. So they're actually very good. If we didn't have the fungi, we'd be up to our eyeballs in dead things. And so it's good to have these guys around so that they keep these things from building up. They're a good source of antibiotics. We're going to talk about that in chapter 12. Also, alcohol, organic acids, and vitamins. We get a lot of these different things. If you like to drink beer and wine, that's where you get it from the yeast, so they're beneficial. And again, making foods and using them in genetic studies. So again, lots of different things. So fungi are not all bad. Some are actually very beneficial. We use these things, and obviously some can cause disease. Just like any other group of organisms, some are harmless, don't do anything to us. Others are parasites, and they live on us, and we have to deal with them. And the same thing goes for the fungi. All right, and here's some of the major lung infections. Again, I'm not going to waste your time and go through all these different things. We'll talk more about these in chapter 22 again, but I just wanted to give you a list of some of these different things. Again, some cause skin issues, some cause mucous membranes, and other things get more deeper into the lung and skin and that stuff. So again, you can look at some of these different things. Maybe you've suffered from some of these things, and you can see what actually causes these things to occur. All right, the next group of organisms are the protists, and the protists are the kingdom Protista, and Protista is basically the junk drawer of the biology world. So if you've taken biology, you may have heard this before, but they're basically the cells that we don't know where they really should fit, and we just kind of shove them into the Protista. Typically, they're the two main types of cells. They're both plant-like and animal-like. There are some that are fungi-like too, but the two main types are the animal-like cells, and then the plant-like cells, which are the algae. And so again, the algae are the plant ones. They photosynthesize, have chlorophyll, just like the green plants and everything else. Whereas the protozoa, these are the single cell organisms. And these guys go in and they basically are mostly free living in ponds and rivers and things like this, but they can cause disease. And so we'll spend a little bit of time in chapter 23 talking about some of the different diseases. I'll show you a few today just to kind of get your wet your whistle a little bit on those things and kind of get you thinking about disease here in a little while. All right, so the algae, again, are photosynthetic. They have those pigments that allow them to absorb sunlight and convert it into glucose, so they don't have to eat. So that's the kind of nice thing about being able to photosynthesize. You never have to worry about eating because all you have to do is be in the sun and the food's made for you, okay? Most of these guys are microscopic. However, we do talk about the big kelps and other things like that. Those are macroscopic and they look like plants, but they're actually algae. And so that's one of those things. One big thing is they all have cell walls like plants do. So they have the cellulose. And again, some have flagella, so they're kind of have a mixture of some that have flagella, some don't. But typically, most are found in the water. And again, that's where we see these things in the water. And again, most of the time, we don't worry about algae. Where we worry about it is when they cause blooms, they release toxins in the water and they can poison the water. They can kill fish, they can kill your dogs if they go swimming in it, and they can kill you if you drink it. So that's where we worry about algae. So when we talk about algae blooms and things like this, if you remember Toledo, Ohio, 2 summers ago, couldn't drink the water for I think it was almost a week, couldn't drink or even bathe in their water because it was polluted with this algae because of the stagnant water and this algae blooms and that stuff. So this is where we worry about algae. Really, anything else we don't worry about algae too much about. Okay, algae are important because they do provide a source of food for a lot of animals. They also produce a lot of our oxygen. So we are happy to have algae quite a bit because they produce a lot of the atmospheric oxygen we need. However, they do produce toxins that can kill you, can produce neurological symptoms and a lot of other things. And so that's the one adverse effect of algae. So algae normally doesn't bother us. We don't worry about it. We see it kind of swampy and that stuff. But typically, we don't worry too much about it unless we're talking about our water supply, or we want to go swimming or other things, and now we're not allowed to because of the algae blooms that are going on. All right, so the next group is the protozoa. There's about 65,000 species of protozoa, and again, these are single-cell animal-like cells, so they have to eat. They are, again, a lot like our cells because they don't have a cell wall. They have the same components in their cells like we do. They have all the same organelles and everything. Most are harmless and swim in the water, and they're fine. And again, unless you look at pond water, you probably don't even know that they're there. The problem is that some do end up being parasites in the human body, and so this is where we worry about some of these things. And I'll bring up some of the cases of the parasites here in just a minute. But that's where we worry about these things mostly, as parasites. Again, they all have to eat like animals because they lack chloroplast, so they have to eat. And again, they have different types of things. And again, they feed on either other microbes or your tissue or organic matter. And so we'll talk more about that as well as we get into the diseases and that stuff. So again, here are some of the diseases. And again, I won't spend the time right now, but we'll talk about these again in chapter 23, looking at these different types of protozoa. Some can be some pretty major diseases like malaria. You probably have heard of toxo, and we'll talk a little bit about this, or toxoplasmosis. This is what we get from changing cat litter and that stuff. So pregnant women, beware, and that stuff. And then again, obviously, Milwaukee's very mindful of cryptosporosis, which is cryptosporidium. And again, that caused a problem with our water supply. And we'll talk more about this when we get into chapter 23 as well. Okay, so some of the different diseases. So some of the ones that are important to know. One is the trypanosome. Trypanosomes are typically carried by insects. So one of them is carried by a black fly in Africa, and it causes African sleeping sickness because the parasite gets into your blood and then goes to your brain, and then can cause different types of things, neurological issues, and eventually lead to death. The other one that's out there is found in Central and South America, and this is called Chagas. And Chagas disease is carried by this Trypanosome cruzi. And this cruzi is carried by this thing called the kissing bug. And what it does is it crawls up on you in the middle of the night when you're sleeping. It goes right by your mouth. It's attracted to your CO2. It bites you in the corner of the mouth and then poops in the wound. And so that pooping releases the infection. You get that infection, and it can cause some serious complications, including enlargement of organs, gets into the blood supply, can cause these different things and other things that can happen to you that can lead to death. And so, and again, a pretty serious disease. If you're going to visit Central South America, you might want to be aware. A lot of times you'll sleep with mosquito nets and that, and one of the reasons why is you don't want this bug landing on you in the middle of the night, biting you and pooping in your mouth. So there you go. So that tells you anything about that bug, but that's how it's spread. Obviously, another one that a lot of people get is this infective amoeba. Amoeba is kind of the cell that kind of crawls around like the blob. And again, it's mostly is associated with dysentery. So a lot of times people will drink this by eating contaminated food or water. They drink it, it gets through the digestive, gets through the stomach, and then ends up in your small intestine. Gets into the small intestine and then causes you to have severe gastrointestinal problems. So diarrhea, cramping, nausea, vomiting, things like this. And so can cause a lot of problems. Sometimes it can be asymptomatic and you don't even know that it's there. And so that can be an issue too, that you're carrying a parasite and don't even know it. What's interesting is estimated that about 10% of the world carries this parasite on them at all times. And so that's a lot of people. So if you think 10% of 7 billion people is, what is that, 700 million? That's a lot of people that have those parasites. So that's something that you want to be aware of. Lots of people carry this parasite. You don't want to have this. And again, think about where they drink and eat and that stuff. So lots of people are infected. Another one that we worry about actually can get into the brain. And so we'll talk about this as well. Get a traumatic injury, either eye or sometimes through the nose, or using those neti pots. And I don't want to scare you if you're using a neti pot, but if you use tap water in it, you got to be careful because sometimes you can get these amoebas that will actually cross the nose and go into the brain and cause a brain infection. It has happened here in Wisconsin. Don't want to alarm you, but it can happen. So just be aware when you're using those neti pots, always use saline and that stuff, and just be careful swimming in stagnant water because you get any of that stuff in your nose or in your eyes, that can lead to an amoeba going into your brain. You don't want that because that means death. All right, some of the other ones that are spread. So probably the biggest disease that is out there is Plasmodium. Plasmodium causes malaria. Lots of different symptoms, and again, spread by mosquitoes. This is a really nasty disease, kills more people by a parasite than any other disease in the world. More people get infected every year by this than any other parasitic disease, and so can kill a lot of people over a long period of time. And so again, there's been no way to control it. There's no vaccines. There's really only one drug, quinine, that's out there that actually protects people and that stuff, or not really protects, but is used as a treatment. But otherwise, you just have to keep the mosquitoes away from you. The other thing that's out there is toxoplasma. Toxoplasma is found in the intestinal tracts of your kitty cats. And so when your cats poop, they poop out this toxoplasma, especially if they're outdoor cats. If you go and change it, you breathe in those litter granules and other things, the dust. If you've owned a cat, you know how the litter goes everywhere and the dust travels everywhere. Well, you ingest those cysts and things like this when you're changing the cat litter. Doesn't really affect you, but if you're pregnant, can affect your fetus. And so this is why your doctor tells you don't go changing kitty litter, because you can get toxoplasma and it can kill your infant or kill your fetus before birth, because it can cross the placenta and go in and kill the fetus. And so again, that's why your doctor says if you have a cat, no changing kitty litter once you are pregnant, because you don't want to risk getting toxoplasma. Okay, so one of those things out there that you should just heed the doctor's advice on that case. He's actually worried about you. All right, and then the last group that we're going to look at is the worms. And so these are the parasitic helminths. And again, these are big animals. These are multicellular. Again, when you see some of these pictures I show you today, you're going to be like, wow, those are huge worms and that stuff. So I want you to kind of keep an idea on this. And people have these all the time. They parasitize host tissues. They get into the intestines a lot of times, and then burrow out of the intestines and go to the different organs. Some stay in the intestines and then digest using your digested foods and that stuff. Some have very well developed sex organs that produce thousands and thousands of eggs and sperm to make new worms, and then you poop those out every time. And then again, go through and then leave the host. So then they go through and they find more people to infect. And so these are pretty nasty little things. Okay, again, we can classify them on the type, the structure, the shape of these things. And so we'll look at these again in chapter 23 and talk about the worms. And again, microscopic detection of worm larvae or eggs in the fecal materials. A lot of times how we determine this, sometimes they're in the blood, other things, sometimes they get into the tissue and they form cysts and things like this. And again, we'll look at this all in chapter 23, but I just want you to be aware of some of these different things that are out there. These are all eukaryotes, very large animals now we're talking about, and again, cause a lot of different problems and infections that can occur. Again, about 50 species parasitize humans. It can be worldwide. A lot of times you see it's associated with dirty water. So a lot of these can pass through fecal oral contamination, meaning that you have poop in the water or poop in the food type of thing. And so that's how it gets into the food and it does affect billions of people, because lots of people in this world don't have very clean water to drink. And so you're using the same water to drink as your toilet, you get problems. And so that can be an issue there. And that's why we see so many of these diseases worldwide. Okay. One of the interesting worms that actually affect the United States a lot of times is called pinworm. And pinworm is an intestinal worm. You can see here, these are what they look like. This is about the size, they look like grains of rice. And so you probably never want to eat rice again. But these worms get into the intestine, and what they do is they come and they lay eggs outside the rectum. And so what happens is the female will move down the large intestine and then lay the eggs outside on the anus. And what that does is it causes an itching sensation. The little kids will then itch their butt. They pick the eggs up, they get them under their fingernails, and then they either grab things and touch things. And what do little kids like to do? Put things in their mouths. So they get inoculation that way. Or the kids will self-inoculate and stick their fingers in their mouth again after itching their butt. And so you get those worms back in, and so it can be these things. Where do we see these? Lots of times in daycares. So daycares can be shut down for pinworm. You may hear this. It is a worm. It is an infection that does happen here and can happen quite a bit. And so if you have any kids in daycare, you want to be aware of this, because it could happen. I'm not saying it will, but it could happen at some point in time. So just be aware of those things, that worms do happen here, even in the United States as well. So we've done a very nice summary. We've talked a little bit about eukaryotic cells, where they came from. And again, the biggest thing is knowing they have a nucleus, they have the organelles. They have the two types. They have the membrane organelles, which are basically your rough ER, smooth ER, and Golgi. And then they have the energy producing, which are your mitochondria and chloroplast. And then they have some other structures on the inside, but really the organelles are the big thing this time. Okay, talk about the flagella. We talked a little bit about it and how they're different than the prokaryotes. Eukaryotic flagella move like a whip. Prokaryote spin. So remember that. I ask questions about that on the test. Okay, so make sure you remember that. And then the internal structures, we talked a little bit about the different types of internal structures again with the organelles, and I talked about that already. The membrane-bound organelles are the rough and smooth ER, and then the energy generating, which are the mitochondria and chloroplasts. And then finally, I finished up with the survey, and again, there's not going to be questions on the test about the different types. I was just kind of getting you prepared, thinking about the eukaryotic cells that we're going to see at the end of the semester. So when we get into the end of April, early May, and we start talking about it, that's when I want you guys to remember that stuff. But we'll talk a little bit more about those things later on when we get further in the semester. But remember, these are the eukaryotes. Most of the time, we're going to spend prokaryotes, but do cause disease. And with that, we've made it to the end of this chapter. If you have any questions, feel free to email me, stop me in lab. We'll talk about it in there, ask any questions that you have on these things. Otherwise, do your Learn Smarts and everything else and keep going. Thanks for watching, and I'll see you next time.